Now that we looked at the structure of muscle tissue, structure of the muscle cells, and most importantly, structure of the sarcomere, we have to look a little bit closer at how we bring about muscle contraction, the mechanisms that are at place, both regulatory and excitatory. So we have to look at the mechanisms by which the sarcomere undergoes this sliding filament theory and how that contributes to overall muscle shortening. We have to look at the roles of both calcium and ATP. We're going to look at calcium on how it plays a regulatory role. Calcium being released allows the opportunity for muscle to shorten. ATP is what's going to provide the energy for that muscle to shorten. So again, remembering what we looked at with regard to our microscopic anatomy, we had our myofibril, and in our myofibril, we had all of our myofilaments that when we got in and looked at closely, took on this overlapping arrangement of actin and myosin filaments, suspended in between these Z-disc proteins, and together we call this our sarcomere. The sarcomere is the smallest subunit of contraction that we have in that muscle. And end to end to end to end, as we go along our myofibril, we are going to see these sarcomeres linked together like boxcars on a train. So as we shorten, each individual one shortens just a little bit, and we can get significant overall shortening of that muscle. As we undergo the sliding filament theory, that is how our muscle is going to generate tension and force, through the actions of the actin and the myosin. Using ATP to drive their sliding is how we can shorten the muscle and generate a force. Remember that when the arm is more in a resting length, we can picture the sarcomere in this manner of orientation. And as long as the actin and the myosin are able to interact with one another, and as long as we have ATP present, then we will witness the filaments sliding over one another and the Z discs approximating. So if we go back and forth here a little bit between this relaxed position and this 50% shortened position, we can see how the myosin sits there in the middle of that sarcomere. And what it's doing is it's using its myosin heads to grab hold of the actin and pull each actin filament, as we're looking at the diagram, one set on the left, one set on the right, pulling them towards the midline, that M line of the sarcomere, thereby pulling each Z disc closer together. You can see that our different bands, our H zone, our I bands, can change in dimensions as we're sliding those filaments over one another. And of course, we can take it one step further and shorten even more, getting an even greater degree of sliding and overlap as we lengthen and shorten that muscle. So now we've gone over that already. Now let's look at how we bring about the sliding. We said before that every single skeletal muscle needs a neuron telling it what to do. We even looked at the arrangements that we see with that with respect to our motor unit. The point at which a neuron innervates a muscle is called its neuromuscular junction. It's a modified portion on the sarcolemma. Now that's where the axon of a neuron is going to butt up against the sarcolemma of a muscle cell, and we see some very distinct anatomical features with that neuromuscular junction. It's got to have a neural component and it's got to have a muscular component, just like the name implies. Neurologically, we are going to have our synaptic knobs, or our axon terminals. Remember, that was the very end of an axon that contained vesicles full of neurotransmitter. Now, we used that term earlier in a very vague sense. We said that there were several different types of neurotransmitters that could have varying degrees of effect on either a postsynaptic neuron or an end organ. In this case, because we're dealing specifically with muscle tissue, the neurotransmitter in question is going to be acetylcholine. So those are our neurological components of our neuromuscular junction. We obviously have to have a muscular component. We have our motor end plate. This is the specialized portion of our sarcolemma that is meant to accept the acetylcholine from the axon terminal. On the motor end plate, we have specific acetylcholine receptors. In the presence of acetylcholine, these receptors open up channels and allow sodium to enter the cell. The space in between, because we got our neuro component, we got our muscular component, the space in between is going to be our synaptic cleft. Now, we used a very similar term before with the synapse, with one neuron to another neuron, and the space in between. This is simply a space in between neuro and muscle. And associated with this entire neuromuscular junction is going to be an enzyme, acetylcholine esterase. We want to have the ability for a neuron to bring about a muscle contraction and then have that muscle contraction end just as abruptly. We want to be able to dump acetylcholine into that synaptic cleft and get a response back. But then we want to stop that response just as quickly. We use our acetylcholine esterase in that neuromuscular junction to remove any acetylcholine in the cleft to stop its effect on the motor end plate of the cell. So we can trace the sequence of events through this neuromuscular junction along the membrane of the muscle cell, the sarcolemma, and down into the inner workings of that cell so that we can see how we bring about muscular contraction. The first thing we have to do is have an impulse releasing acetylcholine into the synaptic cleft of the neuromuscular junction. This acetylcholine is going to open up our acetylcholine receptors, allow sodium to enter the cell, changing the membrane potential of that muscle cell. Remember, we talked about sodium going into and out of a neuron, changing its membrane potential. If we allow sodium to enter the muscle cell, we are also going to get a change in membrane voltage, but this time in the muscle cell. That change in voltage will move along the sarcolemma, and it's going to meet these transverse tubules. Remember those extensions of the membrane that extends deep into the cell? Down those transverse tubules, we have calcium voltage-gated channels. Calcium enters the cell, results in calcium being released from the terminal cisternae and the sarcoplasmic reticulum into the sarcoplasm. Calcium is going to bind to one of our regulatory proteins, troponin. Remember, that was associated with the actin. Troponin is bound directly to our other regulatory protein, tropomyosin. When calcium binds to troponin, troponin undergoes this three-dimensional shape change. And as it undergoes that shape change, it tugs tropomyosin away from the actin filament. What this does is it exposes the binding sites on the actin. Now we have our myosin heads present, ready to bind on to the actin filament. We have our actin filament present with the binding sites exposed. And from there, we got the opportunity now for myosin to bind to actin. Once that occurs, all we need is ATP. And as we go through the process of breaking down ATP into its constituent parts, now we can see this cyclic series of activities where the myosin will attach to the actin, it will pull on the actin, it will detach, it will move to the next binding site on the actin, reattach, and pull again. And it will pull, relax, reach, pull, relax, reach. Not that much different if you picture a tug of war rope, and you got both hands on it. You're going to pull the rope towards you, let go with one hand, grab further up the rope, pull again. This time you let go with the other hand and reach further up the rope. So you're going hand over hand over hand. So one hand's always on the rope while the other hand's always repositioning for another pull. That's an analogy on how this myosin will interact with the actin to get the two filaments to slide over one another. So let's look at these now in more of an illustration manner. We got a couple of different illustrations here, each one focusing on a specific part of what we're looking at. Here is our neuron. We got an impulse coming down the neuron towards the axon terminal and releasing acetylcholine into the synaptic cleft. It's going to bind with our acetylcholine receptors. We see ions enter the muscle cell, therefore changing its voltage, and we get an electrical change move along our sarcolemma and down our transverse tubules. As that impulse is going down the transverse tubules, it's going to meet calcium voltage-gated channels. Similar to the voltage-gated channels we experienced when we did neuro. We need a certain threshold for them to open, and then once they do, in this case, calcium enters the muscle cell. When that calcium enters the muscle cell, it's going to bind to ryanodine receptors on our sarcoplasmic reticulum. And when that happens, calcium is released from the sarcoplasmic reticulum and into the sarcoplasm. Now, that's an awful lot of calcium right there and then. Calcium entering the cell through its voltage-gated channels. Calcium then leaving the sarcoplasmic reticulum. That series of events right there we call calcium-induced calcium release. Calcium enters the cell by way of our voltage-gated channel. It binds to our ryanodine receptors, and that little bit of calcium that enters the cell triggers a massive release of calcium from our sarcoplasmic reticulum. The calcium that enters the cell induces the calcium release from the sarcoplasmic reticulum. Remember, calcium has a regulatory role. Calcium interacts with our regulatory proteins. That calcium binds to troponin. Troponin undergoes a three-dimensional shape change, and when it does, the tropomyosin that it's attached to gets pulled away, exposing the binding sites that are on our actin filament. This being the myosin with the myosin head just waiting to form a cross bridge with that actin. Once we get a binding site exposed, now we can undergo our cyclic excitation contraction. This is cyclic, so we could really start anywhere, but let's start where most of the diagrams have us start, with the myosin attached onto the actin filament. the first thing that happens is an ATP enters the mix and binds onto the myosin head. Now, this first part is a little non-intuitive. When ATP binds to myosin, it breaks the bond between actin and myosin. Now, we get so used to saying ATP is present and ATP does this and ATP does that. Remember back from our earlier discussions on where energy is stored in an ATP molecule. It's only stored in the bonds. We do not get any energy released from ATP until we finally break it down. So when an entire molecule of ATP comes into play here, and we haven't broken it down yet, technically, that's causing a relaxation. We're breaking the bonds between actin and myosin. This becomes significant with certain terms that you might have heard in the past, called rigor mortis. Coroners and pathologists can find a deceased person and time the time of death based on the state of the body. There's a period of time post-death where the body depletes its localized stores of ATP. But obviously, because that person's no longer breathing and no longer undergoing respiration, it can't produce new ATP. Without whole molecules of ATP, we cannot break the junction between actin and myosin. At that point, that muscle cannot shorten, it cannot elongate. That body is essentially frozen in that position. Rigor mortis. ATP breaks the bond between actin and myosin. Now, when we take that ATP molecule and break it down into ADP and the phosphate, we reorient our myosin head. Initially, the myosin head was oriented at this binding site. That's the one that just broke off. When we break down our ATP, our myosin head repositions. It reaches further up the actin filament and orients itself towards the second binding site. When the phosphate group comes off, loses its junction with the myosin head, we get a reattachment of the myosin head. So look at what just happened. The myosin head was attached onto binding site number 1, here in the top of the diagram. ATP comes in, it releases that bond. We break down the ATP, the myosin head repositions, and then ultimately reattaches onto a different binding site further up the actin filament. That was the equivalence of you holding your tug-of-war rope, letting go with one hand, reaching further up the rope to grab hold again. Now, when the ADP portion of the molecule breaks off, we undergo what's called the power stroke. And with that, the myosin head flexes back to its original angle, but this time, because it was attached to the actin filament, it drags the actin filament with it. Now we can go back to step 1. A brand new ATP comes in again, breaks the bond between actin and myosin, and ultimately now we're going to try and orient ourselves at the third binding site. And we will just walk those myosin heads along the actin filament from one binding site to the next. We want to elongate, we do it in reverse. So now we see calcium play a regulatory role in how it interacts with troponin and tropomyosin. If we don't have calcium present, it doesn't matter how much ATP we have, because the myosin head cannot get access to the actin filament binding sites. So we need calcium to regulate whether or not a contraction can happen. Then we use ATP to drive our cyclical, contractile, sliding filament theory. So it's going to be important for you to understand the anatomy of the sarcomere, but also be able to look at the sequence of events. all the way from the actions at the neuromuscular junction, along the sarcolemma, down the transverse tubules. We get calcium-induced, calcium release from the sarcoplasmic reticulum. Calcium ions interact with the troponin, removes our regulatory proteins. Then we use ATP to drive our contraction. Look at that as a sequence of events, and I encourage you to study it in that manner. That way you can fill in the gaps if you start to forget certain items on an exam. So that's going to be the extent of our discussion of muscle tissue in lecture. You will go on and look at muscle naming, muscle structures, slides in the lab. We are not going to include that material in our lecture component. And as usual, if you have any questions on this material, please reach out and let me know. Take care.